Mhm.
Okay. Uh, Good morning with everyone. Good morning, Dr. Stein. Um, Today I'm going to make a presentation about the microbiota, gut and brain axis, a very important axis in the nervous system to regulate the blood, not just the blood-brain barrier, but all the parts in the implications between the microbiota and the neurological system. As you can see in the image, there will be a lot of interactions between the microbiome of the gut and the neural system right in the brain. Like the important thing here is the autonomic nervous system modulation with motility, secretion, permeability, and microbiome. Motility because where when there is changes in the gut microbiome, there are increased motility in the intestine and more secretion, not only of substances in the gut lumen, but substances like neurotransmitters that going back to the nervous system to the brain to regulate the permeability of the blood-brain barrier. That's an important thing that microbiome can do between that two ways. And important part is just to say that it's not only the secretion of neurotransmitter. There is secretion of cytokines. There is secretion of other pro and anti-inflammatory mediators. So it's important to recognize the significance of this system in the regulation, the immune regulation between the gut and the brain. We got to understand that there is a lot of investigations and now confirmed investigations that had put the brain microbiome gut axis in a position as an explanation of a lot of diseases, autoimmune diseases, vascular diseases, depression, and changes in mood because the regulation of this can cause changes in neurotransmitters and the permeability of the brain blood barrier. So we have to say that the relationship between the gut and the brain is bilateral. It's bilateral and bidirectional. There are interaction regulating the key physiological and homeostatic functions, including food intake, immune regulation, and sleep. One of the first studies that we had in this field was investigation from Sudo, showed that the absence of a normal good microbiota, recognized as lactobacillus or streptococcus that is normal in the gut, in early life has significant effects on stress responsiveness in the adult, and that these changes can be partially reversed because if we change that microbiota composition, we can change the response in some adults. So, studies with germ-free animals and specific pathogen animals had, in fact, confirmed this theory, revealing that adult rats that undergoes in changes in their microbiota have different responses in adult life when they grow up. So studies with different bacteria and the modification in course of development in a variety of neurological alteration have been done too. Here is an image of the connection, the direct connection. So most of the people will think about it, how how is connected the gut with the brain if it's in two different parts. Well, one of the important things that connects the brain and the gut is the vagus nerve and the sympathetic nervous system. The two of it are part of the autonomical nervous system. We have to acknowledge that vagus is not only just a nerve, it's the pathway, is the freeway between the gut, between the all other parts of the thoracic and abdominal part of the body, that connects directly with the brain. We got a sympathetic nervous system in the vagus, that we have root ganglions that and plexus that going to make this connection between the gut and the brain. So when there is a microbial metabolism, there is microbiota, and there is small chain fatty acids and neuroactive molecules in the lumen of the intestine. There is an interaction between these metabolites with the cells of the gut epithelium. Well, we got acknowledge that we got neurons directly um, connected to the enteric plexus, to the submucosal plexus, and the mesenteric plexus inclusively, that got neuropods that connects directly with the intestinal epithelium. Dendritic cells that are immune cells that are in the lamina propria or near to the epithelium or the lumen of the epithelium are connected with the neuropod too. So these neuropods and dendritic cells get the signals from the lumen of the intestine with microbial bial metabolites that going to send signals directly to these neurons by these neuropods. So this connection will be significant to get the message through the through all of the way of of the vagus nerve and the sympathetic nervous system. When we get response from the brain, it's get down from for the vagus nerve. um the response in the epithelium is in the systemic circulation, because some immune mediators like tumor necrosis factor alpha and EL8 and EL10 will be released, and it will be transported to the lumen and the cells near to the epithelium of the gut. So this can have a significance between some brain diseases, some neurological and psychiatric diseases, and motility diseases in the gut. Like we got constipation or diarrhea or inflammatory bowel disease, bowel syndrome. And in the brain, we got anxiety and depression that maybe sometimes it's were um, difficult to treat or unresponsive to treatment, because the cause of this anxiety and depression is not just only in the brain, in neurotransmitters in the brain. It's a dysregulation in the microbiome in the gut, truly. So multiple direct and indirect
You're
pathways
frozen.
exist to. Uh, can you hear me?
Yes,
Okay. Can you see the presentation?
yes.
Okay, thank you. Well, multiple direct and indirect pathways exist through which the gut microbiota can modulate the brain gut axis. It's included the endocrine system with cortisol, immune cytokines like the cytokines that we have seen, like the IL-8, IL-1, IL-10, tumor necrosis factor. And there is a neural system, a neural pathway, the vagus nerve, the enteric nervous system, and the spinal nerves, like the sympathetic chain. So several gut microbiomes are capable of synthesizing neurotransmitters like GABA, gamma aminobutyric acid, noradrenaline, and dopamine locally, which can act on target cells in the gut and act as an important avenue of communication between the brain and the gut. So neuroactive microbial metabolites can modulate brain and behavior through a number of ways that are still being elucidated. But now we know that the most confirmed way to affect the brain from the microbiota is the permeability of the brain-blood barrier to metabolites that can activate the microglial system and make changes, immunological changes in the brain. So these include affecting epithelial cells to impact barrier function, and then the enteroendocrine cells to release gastrointestinal hormones, as well as dendritic cells to modulate immune function. We've seen the dendritic cells in the past image, and we know that they can modulate not only the immune function, but they can modulate the communication between the neuropods of the submucosal plexus and the epithelial gut and the gut lumen. The neuropods that we have seen in the last picture have been shown to transduce sensory signals from the intestinal milieu to brain through forming synapse-like connection to afferent nerves in the submucosal plexus. So we got to say that like there is a metabolic input and there is a microbial signals in these things. The the metabolic input in some systems, there is a biosynthetic precursors that got on a multi-step biosynthesis and protein synthesis to get MAMPs and toxins. The MAMPs, there is a microbial sign or signals that get like interaction with the system, the nervous system, like the toxins that can make contact with nervous systems too. Now, in the side of primary bile acids, they can do the conjugation and the hydroxylation to get secondary bile acids. And in other part, cholesterol can do active hormone metabolism. And the other part is the diet. The diet is so important to modulate the microbiome of the gut because we get a lot of the microbiome from the diet. So amino acids can get neurotransmitters, indoles, polyamines, phenols. So if we got a diet that is not the correct diet or the best diet, we got troubles in the composition of these neurotransmitters. So polysaccharides and polyphenols get other part of the system of nutrition in the gut that are connected to the brain. But the important part of this picture is to show the short chain fatty acids that we're going to review it later. So you got to know that there is a lot of signals, neuroimmune signals. The immune system is very connected to the neurological system because the neurological system can control all the body between the immune system. Like the immune system is like a mediator between of the brain for all the body because the immune system is in all the body. Is pleiotropic. So uh, mouse models of multiple sclerosis and stroke have identified substantial roles for gut microbial regulation of autoimmunity, inflammation, and immune cell trafficking. The good microbiota influence the development and function of the CNS resident immune cells, especially the microglia. As I said before in other picture, the connection between the gut and the brain is the blood barrier. So a lot of toxins, a lot of neurotransmitters, a lot of cytokines get to the brain from the gut and enters because this alteration in permeability of brain-blood barrier. And the first cell that will encounter this and get affected by this is microglia. So toll-like receptors like the 3 and 7, which recognize viral RNA, and toll-like receptors 2 and 4, which recognize peptidoglycan and lipopolysaccharide of the bacteria, are activated by microbiota. And this is one of the first connections between the gut and the immune system that's going to be the mediator from gut to brain. So if we got a picture where we can explain some of this. The microbiota is in all the gut, but sometimes if there is troubles with diet, if there is inflammatory bowel syndrome, if there is irritable in bowel syndrome, there will be a dysbiosis that would break the intestinal barrier. We got another intestinal barrier here in the lumen of intestine to go through the circulations and the neuropods near to the submucosal plexus. So we got a dysbiosis, we're going to get a leaky gut. So the lipopolysaccharides of the bacteria and the RNA of the virus will be translocated and pass the intestinal barrier to get on the submucosal fields. So they're going to get in touch with immune cells, with dendritic cells, with the neuropods, and there will be an cytokines release. Cytokines is going to be released directly to the bloodstream. So there will be a passage of cytokines through leaky regions, first from the gut, and then they're going to travel to the brain. So the passage of cytokines through the leaky regions will be in the brain blood barrier too, and then there will be an active transport by cytokine specific transport molecules on brain endothelium. There will be a second messenger signals from the endothelium lining of the blood brain barrier, with an excess production of cytokines, and we will have an entry of activated monocytes from the periphery into the brain, because we have to remember that the brain blood barrier is disrupted. And finally, we got a transmission of cytokine signals by afferent nerve fibers like the vagus during infections. So there is another way to get to the brain from the gut. That is the direct neuronal signaling. Receptors like FXR and TGR5 are expressed in brain neurons, but bile acid concentration are low and undetectable in these tissues of healthy subjects. But we have to acknowledge that FXR and TGR5 are more in bile signaling, like in near to the gallbladder, near to the gut, and these receptors are in the brain too. There have been acknowledged that the brain and some part of the nervous system have these receptors, FXR and TGR5, that respond to signals in the gut, but they're in the brain too. Several studies have localized the GPR41 FFAR3 receptors to the superior cervical ganglia, prevertebral ganglia, submucosal and mesenteric ganglia neurons, sympathetic ganglia of the thoracic and lumbar sympathetic trunks and vagal ganglions, suggesting neuronal activation by microbially derived short chain fatty acids. We talk about the short chain fatty acid as a product of metabolite getting from the microbiota, and there's one of the things that uh, activates these receptors in the brain and in other parts of the nervous system, like the cervical ganglion and prevertebral ganglia of the autonomic system. The other part to get to the brain from the gut is the endocrine signaling. Well, this communication is mediated by several microbially derived molecules that include a short chain fatty acid again, the secondary bile acids, and the tryptophan metabolite. The expression of the FXR, the farnesoid X receptor and nuclear receptor, is activated by bile acids. As I said, we don't have these. We have these receptors in a lot of places, but they only normally, as we acknowledged in the past, they response to the bile acids. But now we acknowledge that in the brain there is this receptor, and well, in the brain there's no bile, but they they respond to another. Hi, doctor, can you hear me?
Yeah.
There is a trouble there with Zoom.
Oh, what's happened here? Your screen is gone.
Yeah, let me.
This is an amazing presentation. I'm really fascinated by what you're doing. Really.
Thank you. I'm going to share my screen again. Sorry.
I'll try to edit this if I can.
Yeah.
Hang on.
Let me say my connection is unstable.
So it says you're starting to share, but you don't see the share. Okay. Good.
Okay.
Gonna make it bigger again.
Uh,
The whole screen.
yeah. Wait a moment, please.
Take your time. Please take your time.
Yeah.
Good. Okay.
Okay. I'm going to continue. Okay. As I was saying, there is wait one moment. This okay. There is an
Uh-oh,
uh
went away again.
oh, what happened? The connection is not so good here. Why? Hmm.
I'm so sorry.
Yeah, the connection of the Wi-Fi is not so.
Are you going back home today, or when are you leaving?
Yeah, I'm leaving tomorrow. My friend of Turkey have leave today. He finished a rotation yesterday.
You're back again. Look, if this is not that good, you can do it yourself at home and send me the MP4. Okay?
Okay,
Okay. Look, you and I want this to be perfect. It's a very, very important. This is a very important presentation. I just think it's amazing good, really.
okay. Thank you. Yeah, can you see the?
Yes.
Okay, we're going to continue. So the other part, as I was saying, is like endocrine signaling. The immune expression of this farnesoid X receptor that we thought that it would be only responsive to the bile acids. Now we know in the brain they can respond to not just neurotransmitter, and endocrine signals can be responded by this receptor. So, like the production of fibroblast growth factor 19, it's similarly functioning ortholog FGF1, like um, is similarly functioning ortholog FGF1 5 in mice, which can enter the systemic circulation and cross the blood-brain barrier. This crossing of blood-brain barrier is important to acknowledge that again, that the blood-brain barrier is affected. The permeability of the blood-brain barrier is affected. TGR5, the other signal that we had acknowledged in the last in the past slides. Um, Gut signal is controlled glucose homeostasis by mechanisms including increased glucagon like peptide 1 released by these L cells. So, the other pathway between the gut and the brain is the enterochromaffin cell signaling. Short chain fatty acids and another eight metabolic acids derived from spore forming bacteria of the gut, like Clostridium, regulate the a significant percentage of enterochromaffin cells, 5 synthesis and release. Gut microbiota contribute to the peripheral availability of tryptophan because, as we acknowledged in the past picture, the tryptophan is one of the amino acids, and then as be transformed to serotonin. So, this tryptophan is imperative to the CNS synthesis of serotonin of 5-hydroxytryptamine. Administration of Lactobacillus reuteri normalized the stress-induced behavioral changes and was associated with decreases in circulating kynurenine levels resulting from microbially derived peroxide of hydrogen inhibition of IDO1 messenger RNA expression. So if we get like how can we say it, probiotics, prebiotics like Lactobacillus reuteri, we can normalize the behavioral changes sometimes when it's caused by microbiota changes. We can manage the behavior with this because the connection is real in these studies. So this is another picture to acknowledge the bidirectional interaction between the microvirus, the enterochromaffin cells that produce the serotonin, and the central nervous system affected by the serotonin. Serotonin of 5-hydroxytryptamine is produced by the enterochromaffin cells of the gastrointestinal tract, as we can see in the picture, the green cell. So, with 95% of the body's serotonin stored in the enterochromaffin cells and enteric neurons, and only 5% of the serotonin is stored in the brain, in the central nervous system. So, a principal, not say pathway, storage of serotonin is in the gut. The gut microbiota contribute to the peripheral availability of tryptophan, which is imperative to the CNS synthesis of this neurotransmitter. Like in the picture, we can see dietary tryptophan and mix it to a spore-forming clostridiales, release the short-chain fatty acids and the luminal hydroxytryptamine release to modulate this microbiota. And then this 5-hydroxytryptamine is upregulated too in the enterochromaffin cells and get to the neuropods in the submucosal plexus and gets through the brain in the vagal spinal signals to the CNS. And the modulation of the brain from the brain to the gut gets by the autonomic nervous system modulation, the sympathetic chain. So this is like a two direction pathway. Like the pathway of the vagal nerve goes from the gut to the brain, and the sympathetic chain goes from the brain to the gut. It's like a freeway between Washington and New York, like this. But it's important to acknowledge this connection between these two systems. The other way is that the gut microbiota can communicate to the brain indirectly too, not just directly. Indirectly too by the gut-derived molecules acting on different vagal or spinal nerve endings, or directly by a microbe-generated signals. Alteration in the gain of this bidirectional interaction is in response to perturbations such as psychosocial and gut-directed, like diet, medication, infection, stress, can alter the instability and behavior of this system, manifesting as brain-gut disorders. That is the new name for these disorders or these diseases, the brain-gut disorders. The brain connectome got stress reactivity, sometimes mood changes, sleep changes, visceral sensitivity, and are connected to the gut-derived molecules like neuronal, immune, and neuroendocrine modulators. As we could say, we have seen in the other slides. There is a connection between microbe-derived molecules and gut-derived molecules with gut connectome in immune neuronal and neuroendocrine cells. And there is an autonomic modulation, again, motility, secretion, permeability, and microbiome. Well, gut bacteria in the intestinal microbiome produce a short chain fatty acids, as I said, that directly stimulates the tryptophan hydroxylase 1 receptor, resulting in the synthesis of serotonin or 5-hydroxytryptamine and secretion from intestinal enterochromaffin cells. Vagal afferent signals to the nucleus of the solitary tract and the dorsal raphe nucleus that are important in the serotoninergic pathway in the brain are important, and the latter of which houses the majority of the brain 5-hydroxytryptamine neurons, the serotonergic pathway. So we have to acknowledge this. That connection is directly in this pathway of serotonin to the nucleus of the solitary tract and the dorsal raphe nucleus near to the brain in the pons. So, as we know, serotonin is an important modulator of emotions, of the depression, stress, and not only that, that regulates other pathways between the neural system, like emotion regulated networks. But here we can acknowledge other things, like it's not only serotonin, it's gamma aminobutyric acid too. It's a glutamic acid too. So if we think about it, there is a lot of neurotransmitters that are regulated not only in the brain. There is a connection with gut. There is a connection with infections. There is a connection between, like we said, the lumen of all the body that is connected to the exterior, and then we can acknowledge that now. This this is another picture that explains how the tryptophan getting in the intestinal lumen regulates the production of 5-hydroxytryptamine and modulated by gut microbes. But there is another important part to see is the kynurenine pathway. The kynurenine pathway is one of the usage of the tryptophan. And this usage of tryptophan can get kynurenine and quinolinic acid and tranilic acid and quinolinic acid. And there is two, the tryptophan producing serotonin. So we got a lot of peroxide of hydrogen. There is a disruption of this kynurenine pathway, and there could be a disruption in the tryptophan production. So if we got a bad microbiome in the gut, we interrupt this pathway. We cannot do a good production of tryptophan. Microbiota will interrupt this. And then if we got a good microbiome, we got indoles dependent of these good microbes. Indole is a great part of the tryptophan production. So it's like a lot of pathways that microbiome can affect the production of neurotransmitters. And as I said, not only serotonin, there is another neurotransmitters that are affected by microbes interaction. So good microbiota can release molecules that can activate the neuroenteric plexus, stimulate brain production of neuropeptides, as well as increase gut blood barrier and blood brain barrier permeability. The brain releases molecules that stimulate the neuroenteric plexus and gut function. The vagus nerve sends orthodromic and antidromic connections in senses. So, if we got a connection between the vagus nerve, there would be a connection, endocrine connection, cytokine connection, and neurotransmitter connection. So we got to acknowledge that brain and not brain, but like pituitary gland can release as like CRH can release in other parts the raphe nuclei and the other neurotransmitter releasers, acetylcholine and serotonin, and it's going to travel all around the vagus nerve to the gut. The gut going to produce neuroenteric plexus activation, and the production of acetylcholine, histamine, serotonin, IL-6 that we know is a very pro-inflammatory um, cytokine and other mediators. And then gut going to respond to this interaction, this message with gut blood brain the gut blood barrier permeability increasing and letting the microbials metabolize to getting the circulation. the immune cells will respond to this, producing histamine, CRH, IL-6, tumor necrosis factor, tryptase, and vasointestinal peptide, vasoactive intestinal peptide. So they're going to be what? Brain-blood barrier permeability increase. So it's a bidirectional pathway. We can acknowledge it now. We can confirm it. It's a bidirectional pathway between the brain and the gut. So here is a resume of the afferent arm and the efferent arm of the pathways between the brain and the gut. Like in the afferent arm, we got a changes of the gut microbiota due to usage of antibiotics. We know that using strong antibiotics like clindamycin in a lot of time, we can get Clostridium difficile infection in the gut because the microbiota in the gut will die with the usage of clindamycin, and the Clostridium species like Clostridium difficile will replenish that and produce a lot of toxins, producing a pseudomembranous colitis. And that is one of the examples to explain that the change of the gut microbiota due to use antibiotics and infectious agents or probiotic bacteria will perform an alteration in the circulating levels of pro and anti-inflammatory cytokines and affect brain function. So other part is the modulation of various host metabolic reaction that will have productions of essential metabolites like bile acids, choline, and short chain fatty acids. The generation of neurotransmitters or neuromodulators in the intestinal lumens produce the induction of epithelial cells release of molecules and stimulates afferent neurons, like in the neuropulse that we have seen in the past pictures. The change in tryptophan metabolism can have effects in behavior, and the activation of sensory vagal fibers will have conveyance of information about the state of the intestine to the CNS, to the central nervous system. And from the brain to the gut, we have the HPA axis activation, the hypothalamus pituitary axis. There will be a regulation of immune cell locally in the gut and systemically affecting gut permeability with motility, secretion, and barrier function and gut microbiota composition. In the other part, there will be an anti-inflammatory cholinergic reflex or sympathetic activation with release of neurotransmitters that may affect gut microbiota composition, intestinal permeability, and local immunity. There will be an activation of the CNS regulatory areas or of satiety with impact on the nutrient availability to intestinal microbiota and their composition. Well, there is some barriers in this because nothing can be perfect. So there is some barriers to this bidirectional signaling between the gut and the brain. And one of that is the intestinal barrier strictly separates the gut microbiota ecosystem from the gut-associated immune system, despite their close proximity. But we know that now, when there is this biosis, when there is not a correct microbiome in the gut, this intestinal barrier will be disrupted, and there will be a direct connection between the lumen of the intestine and the gut-associated immune system. So there is the brain-blood barrier function as a regulator of molecular traffic between the cerebrospinal fluid and the circulatory system. The other part is the TLRs on extension of dendritic cells, which trigger cytokine release from these cells, as well as activation of other cells in the gut-associated immune system. We can talk now about the illness like multiple sclerosis. In the past, there was a lot of studies saying that parasites will help to treating multiple sclerosis because the interaction with the microbiota of the gut and the interaction between the parasites and the immune system getting an anti-inflammatory release of mediators will help to treating multiple sclerosis. And we can acknowledge that now with microbiota, change it not only the behavior in depression and anxiety, changing the immune response directly, like in illness like multiple sclerosis. So, what is the beneficial function of gut microbiota? Is a defense against pathogen colonization by nutrient competition and production of antimicrobial substances, the fortification of intestinal epithelial barrier and induction of secretory IgA synthesis to limit pathogenic bacterial penetration into tissues, and facilitation of nutrient absorption by metabolizing indigestible dietary compounds, the participation of maturation and functionally of the host immune system by providing diverse signals for tuning the host immune status. So again, we can remember there is a lot of illnesses that are in the brain, in the nervous system, autoimmune diseases that are regulated by the immune system. Well, a dysregulated immune system, and we can regulate this immune system that is getting is harming the brain with changes in the microbiota. Well, talking about neuroinflammation and neurodegeneration, good microbiota homeostasis could be disturbed by Western diet sometimes. Like our diet is not the baddest diet of all the world, but it's not sometimes not a good diet. There can be drugs here in the United States. I can acknowledge that there is a lot of usage of drugs, and increasing age. While the probiotics and dietary fibers is beneficial to the maintaining the homeostasis of the good microbiota, the dysbiosis of the gut microbiota would induce the gut epithelial inflammation together with a tight junction disintegration and the apoptosis of these tight junctions. Remember that in the blood-brain barrier, we got the impermeability of the blood-brain barrier is is possible by the tight junctions. So pro-inflammation factors and primary immune cells from the gut tract would translocate into the systemic circulation, which might be reversible by promptly correcting the gut microbiota homeostasis gut epithelial cells. So senescence of somatic cells could be induced by unresolved chronic systemic inflammation, with activation of signaling pathway like the one described here at the early stage. So senescence is one of the important part of neurodegeneration too, and would be regulated too by the good microbiome. So sustained systemic inflammation would cause the remodeling of the brain blood barrier architecture with increasing permeability and collapse sometimes. Influx of the immune cells and pro-inflammatory factors would activate the microglia in the brain, causing neuroinflammation and neurodegeneration. This is one of the pictures to explain this. The good microbiota homeostasis and good microbiota dysbiosis will going to be integrity impairment of the brain of the gut barrier. Sometimes in diseases like inflammatory bowel disease, and there will a chronic systemic inflammation and inflammation with aging like senescence will affect the blood-brain barrier integrity and we're going to impair this barrier. And there's going to be neuroinflammation, neurodegeneration, changes in the composition of the microglia, in the response of the microglia, and this can explain sometimes multiple sclerosis, like that the millennium process can be a part of neuroinflammation process and can be regulated from the gut. And the other part where is the Parkinson disease accumulation of alpha synuclein, and the other part is Alzheimer disease with tau protein and amyloid beta accumulations is neurodegeneration, maybe dysregulated not only in the brain, in the gut too. The gut will send signals that will cause the neuroinflammation and neurodegeneration and can help in the development of this disease like multiple sclerosis, Parkinson disease, and Alzheimer disease. So brain aging and inflammation is important too. There is a significant transition from the young healthy brain to the age dysfunctional brain that involves an hyperactivation of tumor of transformed growth factor beta signaling in astrocytes. Microglia activation is a typical feature of neuroinflammation and neurodegeneration, which involves the forthcoming of brain aging. Strengthening and pro-inflammatory factors transcription like IL-1, IL-6, tumor necrosis factor alpha, and interferon gamma, and decrease it on the EL10 and transfer growth factor beta that are anti-inflammatory. And the third, the microglial cells are sentinels in the CNS to sense pathogens in the surrounding environment. Notably, pro-inflammatory microglia are suggested to cause brain-blood barrier dysfunction and leakage. One of the things that we don't know exactly about multiple sclerosis is how the blood-brain barrier can permit the penetration of cytokines or pro-inflammatory cytokines to the to the brain, so to cause the myelination of a lot of astrocytes, a lot of neurons in the brain. The oligodendrocytes implied in the multiple sclerosis are affected by the cytokines that are passing the blood-brain barrier, and this is one of the explanations because there is a lot of dysregulation in the cytokines transported from the gut. So there is another picture to explain how the segmented filamentous bacteria, Clostridia bacteria, can disrupt and destroy the tissue homeostasis, disrupted an apoptosis of the intestinal cells. Surface layer protein A can connect it with the circulation, and then there is a pathogenic Th17 activation. There is a Th1 activation, and remember that Th17 activation is a principal pathway to destroy oligodendrocytes and cause demyelination in multiple sclerosis. So they're going to be a blood-brain barrier destruction and the pass of the cytokines to the brain and get autoimmunity to produce multiple sclerosis. So there are going to be a lot of risk factors in the intestinal bowel, in the bowel system disruption. There are going to be a central desensitization, psychological comorbidities, altered brain structures, HPA axis dysregulation, altered response to dietary components in the brain. And in the gut, they're going to be altered permeability, altered motility, increased visceral sensitivity, altered gut microbial composition and metabolism, altered response to dietary components, and altered metabolism of the bile acids. And there, with a bad microbiota, there's going to be more lipopolysaccharides and gram-negative bacteria, more mast cells and histamine released, more bacterial tryptamine, and less epoxanthin. So the diet, the use of probiotics, the antidepressants like SSRIs or TCS, tricyclic antidepressants, and the fecal microbiome transplantation will help to replenish and get a good microbiome and get again the homeostasis between the brain and the gut. So we got gut-brain interactive disorders. There several evidence show that these gut-brain interactive disorders can result from the combination of genetic and environmental factors such as antibiotic exposure, infection, and physical or sexual abuse, resulting in synergic gut and psychological health dysfunction. Post-infectious inflammatory bowel irritable bowel syndrome is a great example of the involvement of the gut microbiome and the pathogenesis of these gut-brain interactive disorders. And is it that characterized by the development of irritable bowel syndrome symptoms following infectious gastroenteritis, with evidence that the new virus in the pandemics, the SARS-CoV-2, can infect the gastrointestinal tract, and it is plausible to wonder whether in this incidence of gut-brain in disorders, and particularly the post-infectious irritable bowel syndrome, might increase significantly in the near future. Remember again, the antibiotic exposure commonly seen after infectious enteritis is known and induced microbiota alteration and appears to be the risk factors for the brain and gut disorders. Inflammatory bowel syndrome patients have been shown to have an increased abundance of firmicutes and decreased abundance of bacteroidetes associated with increased ratio. Clostridia and Clostridiales and decreased Bacteroidea and Bacteroidales, as well as a reduction in butyrate producing and methane producing bacteria, can affect a short chain fatty acid that will travel to the brain too. So to remember and acknowledge that the connection between the brain, the connection to the gut is important to explain. First, to explain behavioral changes, depression, anxiety, and illness, psychiatric illness. But now we can acknowledge that there is a lot of neurotransmitters and a lot of changes in the blood-brain barrier and cytokine and immune changes that can harm the brain and cause and not be the only cause, but be risk factors or contributory factors to the pathogenesis on illness like multiple sclerosis, Parkinson disease, Alzheimer disease, alpha-synucleidopathies, and other autoimmune disease in the brain central nervous system. I can acknowledge now that the brain and the gut are very connected and have an important significance for this in the future. This is my references, and thank you for your time.
Okay.